We'll take some examples of gene therapy. Okay, so first example is of cell-based gene therapy. I will take one example related to the chimeric antigen receptor T cell therapy, that is CART therapy. Okay, so chimera, we can pronounce it. So this is a CART or chimeric antigen receptor based therapy used for acute lymphoblastic leukemia treatment, and it is used for treatment of children and young adult patients up to age of 25 years old with relapsed B cell ALL. Okay, that is acute lymphoblastic leukemia. So now to understand that what is chimeric antigen receptor T cell therapy, we must know what is CART, right? So you know that T cells are very important effector cells of our immune system, and they recognize tumor cells and kill them. Their role is to recognize tumor cells and cells with the marked with the foreign antigen. They screen them and effect a launch an effective function effector response and kill them either by apoptosis or by release of enzymes and other proteins. And also they secrete multiple cytokines to activate other immune cells. So they have a receptor known as receptor complex known as T cell receptor CD3 complex on these cells. Okay, so this TCR CD3 complex is responsible for recognition of the target antigen on the cancer cells or other infected cells. Okay, so now this interaction is MHC dependent. The T cell and interaction of the tumor cell is T cell, MHC dependent. Now, because we want to target the lymphoblastic leukemia B cells, or any, in that matter, any other cells very specifically, because gene therapy must be very specific, right? So we can engineer T cells such that they recognize the target or antigen of our interest. So how can we do that? We can do that by engineering the receptor itself. Okay, so if we talk about a T cell receptor, it has many components. It has not only the receptor, which is itself is made up of many polypeptides and CD3 complex and other co-stimulatory molecules. So there are many things to understand. Okay, that we will be understand taking TCR and CD3 complex in different class. But for time being, you understand that TCR CD3 complex is engineered to detect the specific antigen on the cancerous cells in this chimera gene therapy. So here you can see that this is the T cell and this is the T cell receptor in the figure, where you have a signaling domain and the domain that is responsible for the recognition of the antigen. So any CART receptor has three domains, or in, if we talk about any cell receptor on the cell surface or inserted in the cell membrane, it obviously will have three domains. One will be the extracellular domains, transmembrane domain, and the intracellular domain. So if we talk about this receptor, it has signaling domain and the uh, the domain, extracellular domain, that will recognize the antigen. So what do? We engineer the receptor by taking the signaling domain from the TCR and the recognition domain from the antibody, which is specific for the antigen present on the cancer cell. So now we have a chimeric antigen, because it will enable the identification or recognition of the antigen on the cancer cell, as well as it will be able to carry out the T cell signaling and activate the T cell for its effector function. So first step in the CAR cell therapy is to make a CAR gene, that is making a chimeric gene having the signaling domain of the T cell receptor and the recognition domain from the antibody, because we know that antibodies are very specific in their function. Okay, but they never err. So we take the this antigen recognition domain, or we can say variable region domain of the antibody, and fuse it with the signaling domain of the T cell receptor. Now this once the CAR gene is ready, we prepare a viral vector harboring this chimeric antigen receptor T cell. Okay, so now the this once we engineer the viral vector, it is ready for the transfection of the T cells. So we select the T cells from the patient, either autologous or in an allogenic way. We can take the T cells, and then we transfect the engineered virus into the T cells. Now these T cells will become uh, genetically modified. Okay, so what will happen then? Now these engineered cells having the engineered chimeric antigen receptor T cells, now these T cells will be re-injected into the patient intravenously, so that they can carry out their function of killing of the tumor cells. Okay, now in particular in case of acute lymphoblastic leukemia, the antigen that we want T cell to recognize is the biomarker on the B cancerous B cell, that is CD19. Okay, so this designer or chimeric antigen receptor in the recombinant T cells will now recognize the CD19 very specifically and kill only the tumor cells. Okay, so this is the general outline how that first of all we make a CAR gene, then we produce a recombinant viral vector, and then we transfect the T cells with that viral vector extracellularly. Okay, so you this is an example of ex vivo gene therapy. Okay, then you inject these recombinant cells into the patient where they carry out their therapeutic function. You can see in this figure that a T cell is interacting with the tumor cell, and this T cell is having a CAR receptor, that is chimeric antigen receptor, having the SCFE region from the antibody, that is single chain fraction variable region coming from the antibody. And you can see that there are different colored region in its intracellular domain, right? If you see this figure A, there is a T cell receptor, normal T cell receptor, not engineered one. The T cell receptor does interact with the MSC peptide. Uh, okay, so the antigen is in the peptide form, and this interaction is dependent on the MHC. Okay, so this TCR recognizes the peptide presented by the MHC molecule on the target cell, and then there are other interactions of the T cell and the target cells, like CD27, CD70 interaction, CD28 and CD80 interaction, and 41BB interaction with the 41BB ligand. Okay, so all these interactions are important for T cells to carry out its effector function, for its survival, and to make memory T cells. So if we want to launch a very effective and very long lasting T cell response, then we must have all the interactions or activation of the pathways being activated by all these interactions. So once we plan to design a chimeric antigen receptor T cell, we obviously will have this SCFE region from the antibody, and we may either have only the intracellular domain of only TCR, or we can add some important domain which will be activating multiple pathways from different receptors, like a portion or the domain responsible for the signaling through CD28, signaling through 41BB, and signaling through the TCR receptor. So this way we can have different kinds of TCR, this CAR T cells made. Actually, these are nothing but different generations of CAR T therapies. Uh, just to highlight or underline the few characteristics of these therapies is that in contrast to TCR mediated recognition, that is normal TCR, not engineered one. So TCR mediated recognition, CAR is not dependent on the MHC, first thing. And the CAR molecule will recognize any target on the tumor cell surface, and it is not limited to be a protein, since antibodies can bind to carbohydrates and lipids. See, in case of MHC, we have a extracellular domain that interacts with the MHC peptide. But here we have extracellular domain coming from an antibody V region. So now, because antibodies not only recognize peptide antigens but also carbohydrates and lipids, so we can play with this SCFV region also to specify the target, right? So it is not limited to only protein antigens or protein targets. So we can have a wide variety of antigens to target, right? And then the CD28 is one of the proteins that is expressed on the on T cells that provide the co-stimulatory signal for the T cell activation and the survival. So if we include this CD28, so this way we can enhance the T cell survival and activation. Likewise, 41BB is a co-stimulatory signal for that play a very important role in expansion, acquisition of effector function, survival, as well as development of T cell memory. So here comes the T cell memory. So we want to have a T cell memory, then we must include this region also. And then the CD27 constituent can enhance the survival of the T cell and enhance the anti-tumor activity of these cells in vivo. So having these many domains in the intracellular region of the CAR T cell CAR receptor will be very important. Now I will give you a brief idea of different generations of CAR. There are four generations basically: first, second, third, and fourth generation. As you can see, in the first generation card, the engineered or chimeric antigen receptor had extracellular domain, intracellular domain, and intracellular domain was coming from only the TCR, that is CD3 zeta chain, having the signal transducing domain of the T cell receptor. Now in second generation we had another domain, either CD28, OX40, or 41BB. In case of third generation we added one more, we had two domains. And in case of fourth generation, now we have added a domain that can induce the cytokine production. So this fourth generation is also known as T cell redirected for antigen unrestricted cytokine initiated killing. So what does it do and how is it useful? As we have discussed these three types, this fourth one I will give you a little bit information about that. This fourth generation card can activate and attract immune cells, okay, because it activates the cytokine production, so it can activate as well as attract innate immune systems to eliminate the antigen negative cancer cells in the targeted lesion. For example, there are different kinds of cancer cells in the tumor, okay, for example, and we have a targeted CAR therapy for particular cell population in the tumor, but we are not targeting many other cancerous cells in that lesion. So because this fourth generation trucks will induce cytokine production, so they will attract native or innate immune cells, so those cells will eliminate the antigen negative cancer cells. So this way this therapy is also looks very promising. Another drug used as gene therapy is vitravine that we are taking example of. This is an antisense oligonucleotide. So now we have different type or different altogether different strategy of combating the genetic disease. we can use antisense oligonucleotides, okay, as gene therapeutic product. Okay, so this vitravine particularly is designed as a therapeutic strategy for cytomegalovirus retinitis. Okay, this is a serious viral eye infection in case of HIV positive patients who don't have an option for CMV retinitis treatment. Okay, so we can administer this antisense oligonucleotide in such patients to for treatment of cytomegalovirus retinitis. Okay, so this antisense oligonucleotide is a 21 base pair long phosphorothioate oligodeoxynucleotide, okay, which has, in addition to 21 base pairs, a CpG motif, okay, at its 5' terminal end. And what does it target? It specifically target the IE2 mRNA molecule, which is encoding a protein required for CMV replication. So, because you know what, how antisense oligonucleotide acts, they bind to the mRNA and then they stops the translation of that particular mRNA, right? So this oligonucleotide will target the very essential gene for CMV replication, that is IE2. So IE2 mRNA will do that. It will bind this ASO, this vitravine ASO will bind to this mRNA and will stop the protein production, so hampering the CMV replication and thus treating the retinitis. As we know that p53 molecule is a very important, or I would say critically important protein to suppress the tumor formation. Okay, so if this protein gets inactivated in most of the tumors, they become, you know, in a way malignant, or they become hard to, you know, treat. So if we can replace you know, or if we can give a functional copy of p53 in tumors, then we can treat those tumors. So Gendicin is p53 gene therapy that is mediated by the adenovirus. So we use here an engineered adenovirus harboring a functional copy of p53 gene. Okay, so this drug carries out restoration of the p53 gene by adenovirus mediated gene therapy. Okay, in this drug, even region of the adenovirus, that is human serotype 5 adenovirus, has been replaced with a human wild type tumor 53 copy. Okay, so now this adenovirus is a recombinant virus having the human wild type p53 copy. Okay, so the expression of this p53 gene in cancer cells stimulates the anti-tumor property of this protein. Okay, so it activates the healthy copy or functional copy of the p53 gene now in the cancer cells, will initiate anti-tumor activities, and it will initiate apoptotic pathway, it will initiate autophagy, it will suppress the DNA repair, and do all it will stop the, you know, survival pathway in the cancerous cells. So this way, presence of a functional wild type p53 copy will help, you know, in elimination of the tumor. Okay, so this recombinant adenovirus harbored p53 now give a functional copy of p53 that that activates you know, endogenous angiogenetic inhibitor. Okay, angiogenesis is always, you know, help in the tumor growth. So this wild type copy now activates the inhibitors of angiogenesis. Okay, so this way it helps, and it inhibits the different growth factors leading to angiogenesis. Okay, and it also create you know, microenvironment that helps in arresting the growth of the tumor cells. Okay, it makes the tumor chemosensitive and radiosensitive. It activates the autophagy pathway in the tumors. It you know, activates the pathway leading to the apoptosis in the tumor cells. So this way, replacing or giving a p53 functional protein in the tumor environment will help eliminating the tumor. So Gendicin is used for that. Another gene therapy example is Mucogen, okay, or it is also known as pagaptanic. Okay, so what is it? What is the chemical nature of it? It is a polynucleotide aptamer. So it is based on the aptamer therapy. Okay, and it is used for vascular endothelial. It targets this vascular endothelial growth factor for neovascular age-related macular degeneration treatment, AMD treatment. So what is it? It is a polynucleotide aptamer. And what are aptamers? Actually, aptamers are small RNA, DNA, or nucleotides, or even peptides that targets any antigen very specifically, or any protein or any target molecule in a highly specific manner. For example, here you can see this is a aptamer sequence. It refolds, or it takes a functional form in vivo, and then it targets very specifically to it binds to its target very specifically. So that's why, because of the specificity only, these are very important agents that can be used for therapeutic purposes. So this Mucogen is used for treatment of age-related this macular degeneration, that is an eye disease where the macular region starts degenerating because of the very much growth of the vascular tissue. So actually we want to suppress that over formation of the vascular tissue. So this targets this vascular endothelial growth factor that stops that growth. And this Mucogen is the first anti-angiogenic agent approved by the US FDA in the year 2004, and it was the only therapy for treatment of AMD during that time. And it is also the first therapeutic aptamer with RNA structure achieving the FDA market approval. Okay, now other treatments are also available for treatment of AMD, like different monoclonal antibodies are there. Because these gene therapeutic drugs are highly costly as compared to the other therapies. So nowadays the market is obviously reducing for this product, but still it is useful for treatment of age-related macular degeneration. The next example include Oncodine. This is a very special gene therapy product. Actually, this is the first oncolytic virus. So this is actually a recombinant virus again, approved by Chinese FDA, and it is a recombinant human adenovirus type 5. Okay, and this in this virus, a particular gene, that is E1B55KD, has been totally depleted. Okay, actually this gene is responsible for the p53 inactivation. Okay, so in any tumor, if you inactivate the p53, so the tumor survival and tumor growth will be enhanced, right? So if you delete this gene in the virus and infect the tumor with that particular virus, okay, so what will happen? The virus will not be able to inactivate the p53 gene, but still able to infect the cancerous cells. Selectively, Oncodine propagates in p53 deficient cancer cells, while the adenovirus that lacks this gene, E1B55KD, fails to replicate in normal cells. This is also a very important feature of this drug. So it must not, this recombinant virus must not be able to replicate in normal cells, otherwise those also will be killed by the drug, right? Our purpose is to kill the cancer cells only. So this E1B55 deficient cells will not inactivate the p53, will only replicate in the cancer cells, will not be able to replicate in the normal cells. So these three features make this oncolytic virus a very important drug. Okay. uh, it was initially licensed for the treatment of patients with last stage refractory nasopharyngeal cancer in combination with, of course, chemotherapy following phase 3 trial, phase 3 clinical trials. So once this oncolytic virus is administered following the cancer lysis or cancer cell lysis, what happens? This adenovirus get released in neighboring in cancer microenvironment and infects the neighboring cells and initiate the cascade of cytotoxicity on oncodine mediated cell cytotoxicity, leading to the lysis of the cancerous cells. For we will see that this figure shows you that the mechanism of action of this oncolytic viruses. Okay, so as they have ability to directly kill the tumor cells, and they also have the ability to stimulate the host anti-tumor immune system responses. Like here you have the oncolytic viruses, and once they are injected uh, they only act on the tumor cells. They infect the tumor cells because they are not able to replicate in the normal cells. They replicate, multiply, and lead to the death of the tumor cells. And side by side they activate the innate tumor anti-tumor immune system by activating different immune cells, and by production of the cytokine, this environment become lethal for the cancerous cells. Now once these oncolytic virus kill the cancerous cells, they are released, and they again repeat the cycle by infecting the cancerous cells. So this way selective killing of the cancerous cells is positive by the oncolytic viruses, which are recombinant gene therapeutic vehicles. Okay, so this way we can treat multiple diseases by taking different approaches of gene therapy. Thank you, viewers, for watching this part 2 of gene therapy, and please subscribe, like, and share, and keep coming on my channel.